Hi, teacher Alice. Today we will be presenting our experiment of cellular respiration for our third trimester exam. The participants of this group are Amisha, Bhagchandani, Jaqueline, Sherin, Jaafar, Solmarie, Jaramillo, and my person, Nawal, Elneser. Cellular respiration is the process through which cells convert sugars into energy. There are two types of cellular respiration: aerobic respiration and anaerobic respiration. Aerobic respiration includes the stages of glycolysis, which occurs in the cytoplasm, then the Krebs cycle, which occurs in the mitochondrial matrix, and the electron transport system, which occurs in the inner membrane of the mitochondria. In anaerobic respiration, there's two ways. The first one includes glycolysis and alcoholic fermentation, which both occur in the cytoplasm, or glycolysis and lactic acid fermentation, which occur in the cytoplasm as well. The chemical equation of cellular respiration in the presence of oxygen is C6H12O6 plus 6O2 turns into 6CO2 plus 6H2O plus ATP. Therefore, the reactants are oxygen and glucose, and the products are carbon dioxide, water, and ATP. In this experiment, we'll be testing cellular respiration in plants and which factors affect this process. We'll be using bean seeds, 5 boiled ones and 5 germinated ones, and we'll be seeing how these two different states can affect the process of cellular respiration.
Before beginning with the experiment, you should consider the following lab safety rules and accurate measurement techniques. Number 1. Wear gloves so you don't accidentally spill bromothymol blue on your hands and cause skin absorption of chemicals. Number 2. Always wear your lab coat and safety goggles. Number 3. Handle the glassware carefully. Number 4. Never taste nor inhale any chemical, in this case the bromothymol blue. Don't ingest because it may cause gastrointestinal irritation with nausea, vomiting, and diarrhea. Don't inhale because it may cause respiratory irritation. Number 5. After the experiment, wash your hands thoroughly. And number 6. When measuring liquids like water and bromothymol blue, make sure you're doing it on a flat, even surface.
Materials needed for this experiment will be the following: 2 glass containers with lids, such as baby food containers, each labeled A and B respectively. 10 milliliters of bromothymol blue, which is a chemical that will work as a carbon dioxide indicator. You will also need water, 5 germinating seeds, and 5 boiled seeds. Step 1. Fill 3/4 of each glass container with water. Step 2. Pour 5 milliliters of bromothymol blue into each glass container. Step 3. Take the 5 germinating seeds and place them in container A and put the lid on. Step 4. Take the 5 boiled seeds and place them in container B. Do not forget to put the lid on. Step 5. Record the color of the liquid inside each container in a data table or notebook. Then, let the container sit for 24 hours. Step 6. After 24 hours have passed, check both containers to see if there was a change in color. Write down the observations. Step 7. After writing the results, clean up your space and wash your hands. Step 8. Compare and analyze your results.
After 24 hours, cup A, which was the one that had the germinated seed, changed color from blue to yellow. Cup B, with the boiled seeds, did not change in color and it remained blue. As said before, cellular respiration is a process by which organisms transfer chemical energy from energy storage molecules, glucose, to energy carrier molecules, ATP. This process needs sugars and oxygen for it to be able to produce carbon dioxide, water, and ATP molecules. Bromothymol blue, which is the reactant we use in our experiment, is a chemical that changes color from blue to yellow when carbon dioxide is present. As seen in our experiment, the cup with the germinated seed changed color, which means that the germinated seeds are producing carbon dioxide. So, why is this happening? Seeds of plants have a lot of sugars, and by breaking those sugars down, it releases carbon dioxide as one of its products. The process that is taking place is cellular respiration, which allows a plant to produce the energy necessary for growth and reproduction. However, the boiled seeds do not release any carbon dioxide, and that's because they don't respire, so they don't take any oxygen, resulting in no carbon dioxide being produced.
Overall, all organisms carry out cellular respiration to convert their food, sugar, into the energy needed to have a sustainable life. Cellular respiration involves sugar and breaking it down to transfer energy. Seeds of plants are filled with sugar, and by breaking down those sugars, it releases carbon dioxide. Cellular respiration can't occur in non-living organisms, represented by boiled seeds in the experiment, because they don't have cells anymore. In this case, thanks to the denaturing of the proteins.